So let's start on this presentation. Here we see a model of a coronavirus. We could also look at any other virus model, as these are all artwork. And right away we have the first tough lesson, because none of this corresponds to actual reality. None of it. The proteins, the spikes, the bits of nucleic acid. This is a model that doesn't exist in reality. When you know real biology, this can't possibly exist. Here's the so-called nucleic acid, the theoretical genetic strand of a theoretical virus. That is the heart of the virus. We test that hypothesis here, and the simple thing about corona is that it has never been proven to exist. Out of very short pieces of genetic strands, they calculated mathematically a whole single strand of genetic material that does not exist in reality. Back in history, we go about 2,400 years when this person, Democritus, learned from his teacher and then expanded himself, with motivation to form the basics for our teachers today, for our understanding of life, but also of disease. So Democritus said, we need an explanation of life, an explanation of disease, but we don't want any consciousness, please. We don't want spirit in it. We don't want gods. Why? Because religion always causes fear, and that's why we need a materialistic explanation of life. He postulated that there are atoms that are immortal, and when they join together, they form molecules. Then these molecules merge again, and this somehow results in life. This purely materialistic idea of life sounded great. It was motivation to stop religious fear. But they never imagined that a new religion would emerge from it, namely a religion of good and evil, which forces us to think materialistically. Because if we want to explain something that actually exists, an illness and disease that appear simultaneously or one after the other, we are forced to believe in physical defects as reality. We have no other choice. If talking about consciousness is forbidden, we can't imagine that a word can actually kill, which it can, or that a word can heal. If thinking is forbidden, then it is naturally unthinkable that a word can become or transform flesh, for example, so-called psychosomatics. It was Plato who quoted his teacher, Socrates, saying the Greek doctors can no longer deal with diseases anymore. They were just using medication to suppress symptoms in order to get slaves back to work faster. This man, Rudolf Virchow, was the one who came up with the concept of a defective carrier gene for explaining illnesses. He is one of the most dramatic figures in our history. He comes up with a completely new theory, refuting the old humors, whereby he postulated that disease was caused by disease toxins, because one could observe that one gets sick if one takes disease toxins, like when a teenager drinks a whole bottle of vodka. Up until 10 years ago, they would be dead unless their stomach was pumped out. We all know, well, maybe not all, but Boris Yeltsin needed 2 liters of vodka just to be able to speak in Parliament, and 3 just to get on a Panzer battle tank, because he had antitoxins, as in the old humors theory, where you get better if you have the antidote. That's how this defective carrier gene thing still falsely persists in medicine, of the virus theory and the antibody that is supposed to neutralize it. That is the theory that the majority of the population still believes to this day. And what did he claim in 1858? He said life consists of individual cells. That was clear. Life arises through cell division of a single cell, and all diseases can be traced back to a single cell, producing the disease-poison virus and distributing it to the other cells, and it would then diffuse and the disease would spread in the whole body. And Virchow had political success because he stated that there are individuals in society who are like parasites. He recommended we eradicate them from root to branch, birthing the excuse for eugenics. Once a humanitarian, he injected this theoretical cellular pathology as a scientifically founded claim into the political arena and turned everything on its head, claiming everything comes from the individual cell. So the theory of disease being caused by disease poison would then be official. Virchow hid the information they already had, specifically that disease did not spread. But after 650 years of the magnifying glass, when microscopes came along, it became clear that each organ consists of 4 different tissues. Well, back then 3 tissues were found, but we now know there are 4. So 3 different tissues which, when diseased, only one of these 3 tissues died. But this new finding was not publicized and shared correctly, but instead ignored, so this information was not spread. And if cellular pathology is not correct, then our theory of cells is not correct, and then virus theory is implicated and not correct. Next, we see these particles that are supposed to be the measles virus. They are shown to us and they say these are viruses. Here we see a couple of small parts with circular appearance, spherical, but then big chunks here, and these structures presented to us as viruses have never been seen in humans, never in animals, never in liquids, in mucous membranes, in saliva, in blood, in urine, never. Everything that we are shown as viruses are false. They are just typical tissue decomposition products of the test tubes with which we work with, in which you try to, or believe you can, study life, which goes back to Virchow. He was a pathologist who believed that by looking at decayed matter of decaying tissue, he could somehow learn about life, and that's exactly what we're still doing today. We take tissue from organisms like monkey kidney tissue that lasts the longest in the test tube, at least that's what they like to say, but they really only live a bit longer because we surround them with a blood serum taken from a cow fetus in utero. Serum is blood without any solid components. If we took it from adult cows or grown mammals with no blood stem cells, the tissue would die immediately. But with fetal serum, it stays alive for a while and doesn't disintegrate so quickly. And these signs of decay in these pictures are a unique, specific in vitro test tube phenomenon that has never been seen in actual in vivo life. So quickly we move from being virus believers to an end to virology. These particles here are shown to us as viruses, but what are they really? They take broken down tissue, isolate this tissue from everything around the core that can still survive, and chunks of the tissue develop little fingers, spikes or crowns very quickly, like an amoeba that wants to rebuild the tissue again. That's why everyone who works with these cell cultures and test tubes has to constantly separate them again and again. These little chunks of tissue are misinterpreted as cells, and these little fingers with which it drags itself along to try and rebuild the tissue, these are called the villi, of which a cross-section in the electron microscope underneath is presented to us as viruses. It's that simple. It's hocus pocus zimzalabim. This pseudoscience is the basis of their actions. I can only call something scientific if it hasn't been refuted or falsified. What he means is that in science, you are supposed to set up repeatable control experiments to try and disprove and debunk your own theory as incorrect, not rig up bogus, flawed experiments to make it appear it might be true. If nobody can debunk your theory after valid scientific control experiments, your theory stands as true, or at least plausible. And you didn't do that at any level of your research, yet you paint us these models of viruses that don't really exist. There was an old virology before 1952, and they thought viruses came from protein pieces and that the protein had genetic material. Then the virologist did the same control experiments and determined if we allow healthy organs to decompose first, then do the same experiment, the same filtration steps, we find the exact same proteins that we misinterpreted as viruses. So they immediately carried out control experiments along with attempts at infection and found that no transmission attempts of the so-called infection ever worked. On the contrary, they found that the experiment itself, namely extracts from sick animals injected or dripped into the lungs via a tube, caused illness. That causes the diseases. It has never been possible to transmit an extract to date from a human who was so-called infected. Back then, of course, virology was lacking a virus model. They had refuted themselves, but they established from these phage structures in 1992 that protein cannot come from protein. So protein is not genetic material. But we're forced to think that there must be a hereditary substance. That seems logical. If we believe that life comes about only through cell division of one cell and there is no consciousness, no spirit at work here, then of course we need something to make the molecules work so that from wheat seeds, wheat always emerges, or the elephant or the human, whatever. Then we're forced to acknowledge a hereditary substance that orchestrates the installation and functional plan of life. So it used to be the proteins, then it was the nucleic acid from 1952 onwards, because it was found with research that these proteins that are produced here always come with a piece of nucleic acid as a catalyst. And from 1952 onwards, the DNA hypothesis of nucleic acid being the hereditary substance emerged, and it was believed. Then these new hypothetical viruses took shape, modeled from what we call a bacteriophage, which always has a strand of genetic material inside because that is a fact. It's not surprising what happened next. A bacteriologist named John Franklin Enders, suddenly he takes up biology. He was a researcher and studied everything possible and meets a bacteriologist who worked with phages. He is fascinated, so he immediately takes up biology for his doctorate. Having never studied biology, natural sciences, or medicine, so he was never taught scientifically to check every step of his research. He used the idea that bacteriophage emerged from the tissue of animals and believed that if these tissues die too, it could transform into these viruses. We don't know what they look like, we haven't ever seen any, but if the tissues die in the test tube, that would at least hint that we have a virus, that we have an agent, and he describes what they are doing here. He used this fetal calf blood serum, he uses antibiotics, but never does a control experiment. Otherwise, he would have understood that his tissues are dying because he suddenly deprives them of the fetal calf blood serum nutrients. If he used an adult serum, they'd die too, and the antibiotics alone are already enough to kill the tissues. If Enders had done the control experiments, he would have found that the laboratory materials and technique are what kill his tissues. But instead, he translated this dying tissue into the presence and isolation of a virus as the cause. So finally bringing what was outside into the lab, and we can then repeat that experiment over and over again, and that repeatability is the proof. But they are only repeating a false assumption to this day. And in this publication, which he explicitly described as speculation, he writes, In so doing, however, we must bear in mind that these cytopathic effects, which resemble pathology, could result from a different agent that was already in the tissue or by unknown factors, because we observe that the tissue that we did nothing to also suddenly dies off. Hello? You're studying a process of something dying. Anyway, in a second attempt, an application of a second agent was tried, and the cytopathic effect changes could not be distinguished as any different from the one with the assumed infection. So obviously, the possibility of finding the same effect with other agents has to be considered. He summarizes, accordingly, the results that are summarized here must be subjected to most critical analysis. So that is what we are doing here, giving highly critical analysis. But that never happened. I'll explain right now why. He writes, there is no reason, no assumption, no justification to believe that these results in human in vivo would be the same here as in the test tube in vitro. There is no reason, he writes. We are only dealing with indirect indications, and 2 experiments are essential for the establishment of a connection between these agents that are produced here and the measles virus, namely the infection in humans and animals, that the experiment was supposed to find. Then what happens? John Franklin Enders, 6 months later out of the blue on the 1st of June 1954, for a completely different thing, got a Nobel Prize for medicine. And what came out of this explicit speculation? The Foundation for Modern Medicine arose like a phoenix from the ashes, and the ongoing fight from virologists against the old virus model was lost, as they just made a new one that is unquestionably linked with a pseudoscience technique that has never been controlled. So I controlled their measles virus experiment. Namely, stop the bovine serum nutrient feeding and the poisonous antibiotics, which kill the bacteria that are everywhere and that cause the cell death like clockwork, which then the biologists pass off as a supposed infection as the cells die off, and they say, hey, that's the virus, and based on this false conclusion, production is carried out of this rotting soup for live vaccines. They say the whole infectious virus is still living in the substance. For dead vaccines, only a part of the alleged virus, so just a protein, and more recently mRNA that we call inactivated virus. My control experiment was just doing the exact same procedure, except with no infection, and the tissue dies in the exact same way. Then what does the virologist do? The virologist takes nucleic acid, just little bits and sections, please. He's never seen the whole strand or sequence, like we do with phage, which we have been isolating and characterizing the entire nucleic acid sequence from for 70 years now. We show we always have the same sequence. But the virologist instead constructs something that he calls the virus ABC. And that shows us that this publication is quite clearly taking an anti-scientific turn, and now proves that virology has refuted and debunked itself. They describe step by step that they isolated nothing, that they haven't found anything, and that they had to multiply it a little and then mathematically construct something which you never find in reality. So we do control experiments with nucleic acid from humans, animals, whatever. We do exactly the same thing as the virologists do, and lo and behold, we can construct the exact same genome if we tell the computer program what to make out of the same small fragment pieces. Now coronavirus from the same data, now make the HIV virus. Or here, make Ebola. Or influenza.
You get what you pay for.
Boom. And that's exactly what virologists do today. They look for little fragments, read them, and then build a theoretical strand of genetic material, just as geneticists build a chromosome. So, let's see how corona started. The Chinese tried to calm the wave of panic that broke out at the end of 2019, and Professor Christian Drosten didn't get drunk at New Year's Eve like everyone else, but instead went on the internet, looked at the sequences from the hospital, and thought it's a kind of a SARS virus. He took the sequences attributed to the coronavirus and prepared 3 very different test procedures. So Drosten synthesized the chemical sequence. That alone normally takes 5 days. Yet on the same day the authors of this study posted their sequence, the quote is here at the bottom in Nature magazine on the 10th of January, using the sequences that he calculated from the internet, Christian Drosten sent his test chemicals all over the world. The Chinese government said, look here, we have a sudden emerging respiratory syndrome, SARS, but all 49 cases of pneumonia in the greater Wuhan area were all isolated. All friends, all neighbors, all work colleagues, everyone that they had already been in contact with were all quarantined, and none of them got sick. So this was not SARS. They communicated openly and honestly to the WHO and to everyone saying it's okay, we're handling it. It's extremely difficult to transmit, and we've closed the fish markets and the world markets, and we're not getting new infections, so we can have our New Year's celebration on the 25th January 2020, the Communist Party planned event. Yes, half of China will take to the streets. But what happened? A friend of Drosten's, a doctor, travels on his own initiative from southern China to Wuhan, holds a press conference at noon. All the Western media are somehow already waiting there, and says that the Chinese government is lying. Imagine that. Then, of course, a huge panic breaks out because this doctor says they lied. And look, I have 2 patients from southern China who weren't in Wuhan that tested positive for the virus using the test just whipped up from Dr. Drosten. This is the raw data, and there are these gaps that they fill in with dirty PCR sequences. If I set the PCR to be dirty, then over 14 duplication cycles it will be positive. Then 30 cycles are completely dirty, and 45 cycles are just completely unscientific. And we have shown that there are many sequences that are just typical human sequences. So the panic took off. They can't get this under control anymore. And reading from Fang Fang's Wuhan diary, they brought the panic under control, with military on the streets. And that's how Christian Drosten turned a local panic into a global one. And that's his business model. And that's what he always did with SARS, with Zika. What else did we have in the Congo? They had Ebola. So there we have it, everywhere now. We always hear the same thing. Christian Drosten gives his sequence for the PCR test to the world for free of charge for humanitarian reasons, which gives positive results and gives the impression there's something spreading. For those who believe,